It's always fun to try to figure out what Brother Bowden was thinking. Puts these together with the particular topic for the day. Obviously, doers is the word. Anybody have any idea why doers would be important here?
Talking about the function.
Proteins that perform actions, right? They do things for us. All right? And interesting enough, so in this particular case, doers, not doers only. Okay. So if you're looking at it, if you don't have action, do you have anything? No, we don't. And so as much as the Lord is teaching us that in terms of our personal choices, I think yesterday was pretty good. Think celestial, but that means we also have to act celestial, right?
Yeah.
That's probably the corollary to that. Thinking about it is the first place to go, but then we have to do something about it. All right, so that's where we're going. And we're talking about proteins, and what is it that makes, what is it that determines function? The shape, the arrangement, the molecules themselves, how they're put together, the atoms and where they are in three-dimensional space is what's going to determine what it does. All right? And so that's what we're going to be looking at. We're going to be looking at protein function over the next couple days. All right, chapter 5 material. If you haven't already read it, you should. All right. All right, so the learning goal specifically for what we're doing, we're going to be looking at trying to understand the essence of what a protein does. Most of the time, the protein has to bump into something else, right? They're I don't even know if there is a case, or a protein exists independent of everything else. I don't think it happens, right? If you find one, let me know, and I'm perfectly willing to say I was wrong. Okay, so what we're looking at is often there's going to be some kind of a molecule that's going to reversibly interact with the protein. This is what's called a ligand if you're in biology, all right? Usually it's some kind of a smaller organic molecule. It can also be some kind of a protein that it's going to actually reversibly associate with. All right, so it's the idea that there's something that brings them together so they stick, and then there's the ability for it to unstick. What do you think determines whether two things stick together? The shape. So shape is part of it. What else?
Charge.
The charges. So possible charges that are part of that surface. Anybody else? Okay, so orientation, so that we get the shape in the right direction so that it can actually interface with the protein, the surface of that protein, whatever it may be. All right, so there's going to be specificity of ligands and binding sites, they're going to match up. Sometimes people talk about like lock and key type mechanism, where it just happens to be the right shape to fit in there and do whatever it's going to do. That's okay. All right, ligands often are coupled such that when there is a binding event, it causes a change in the shape of the protein, which then would give it a new functionality that it didn't have before association. It's a very powerful idea if you think about it, especially when you think for one moment about what's really happening. We have a polymer that can spontaneously fold into a given shape, and then it can actually interact with other things based off the properties of the polymer itself. If you're into material science at all, this is pretty amazing when you think about it. Nobody had to go tell it what to do. It was designed and it did it, and it does the job. All right? So it's pretty fascinating in that sense. All right, so if we have this change in conformational shape, that's what's called induced fit. If it's a multi-subunit protein, conformational changes in one subunit can often affect the other subunits. So when you have some kind of multimeric protein, that's what we call cooperativity. So they cooperate with each other. And last of all, these interactions can be regulated. In other words, sometimes you have to have something bind onto one protein before it's going to bind to something else. So there's a possibility of if you will, a logic to it. All right, we're going to illustrate this by looking at hemoglobin primarily. We'll look a little bit about antibodies, and we'll also briefly touch on muscle contraction as an example. All right? All right, with that, where are we going? All right. Function of globular proteins. There it is. Any questions about those? They're just examples. As we go through the next couple of days, we'll look at some of these up here, and then we'll talk about enzymes starting next week. So a slightly different version of what we're talking about today. All right, and those are the biological catalysts. All right. Protein interactions with other molecules. So these can be reversible and transient. It's an okay thing. That's what we call a ligand. They're gonna bind to the binding site, and the ligands will bind to something usually by a non-covalent force. So that's gonna be a charges, ionic type interaction, or some kind of, it can even be instead of ionic, you can have hydrogen bonding. What other forces do we have yet to mention?
Dipole.
Dipole type, so a little weaker than the hydrogen bonding. And ultimately we have our last one, hydrophobic effect. Van der Waals technically describes them all. Yeah, biology loves that word, but it's technically all of it. So not distinguishing. All right, so with that, this is what we see happening. We have our protein, it has some kind of a, what's this?
A binding
site, and it's going to couple with the ligand, and those two will come together. So that's what we call an association. Makes sense, right? And then there's also the possibility of the reversal, because we said it was transient, it can go through a dissociation as well. All right? Now, what's going to determine whether or not this association, that this binds really well? I didn't quite hear it, was it?
Enzyme kinetics?
No, this is just binding. We're not doing any chemistry yet. That'll be next week. So if we just have an association of these two things, what's going to help determine that?
Are you talking about like the binding site itself?
So just start talking to me, yes.
So if the binding site has a uniquely like really good shape for that particular ligand, it'll have a really good like connection between the two.
Okay, so the better the connection, the stronger the association, correct?
Yes.
Well, what's this dissociation about then?
How it pops off.
Yeah, how's it going to pop off if it stays on really well?
I mean, these are not covalent interactions.
Okay, they're not covalent.
Could you also change, like, if something in the environment was to change it, you could also.
Okay, definitely. If there's some kind of a, if it bumped into something else, there could be a conformational change or a slight change that would actually cause it to dissociate. So what we're going to look at is the longer it stays bound, the tighter the association, the longer it'll stay bound, right?
Yeah.
That means the time for it to come off is going to be very long before it would come off. And we're going to be looking at the ratio of the association with the dissociation. This is a dynamic factor or something that we call kinetic, but it doesn't necessarily mean that we're going to be doing enzyme kinetics, because that refers to doing chemistry as well. All right, so the higher the Ka, the higher the affinity for the protein. The higher the Ka, the higher the affinity that ligand has for the protein. Who can reverse that statement in terms of dissociation?
The higher the dissociation, the higher, or I guess the less the affinity is.
The higher the dissociation, the higher the
Lower the affinity.
The lower the affinity, okay. So that's exactly right. So the lower, so we can reverse that as well when we consider the reversal. All right? So kinetics is a process to describe this. So we're going to be looking at the association rate constant and the dissociation rate constant. You might remember from 106, you can write a rate law for it, correct?
Yay.
And if these two are equal, what does that mean?
The equilibrium.
They're at equilibrium. You can define what the equilibrium state would be, whether it's favoring being bound or unbound, right? All right, so looking at that, that's what we have. So of course you can go through, and we're looking at this once we have equilibrium. That's going to be where we have the association dissociation rates. Dissociation rates are equal. And then last of all, the equilibrium composition is characterized by the equilibrium constant Ka. This is big K, not little k. Right? Remember, these are the rate constant. This is the equilibrium constant. If there's anything you got out of 106, it should have been use K for a lot of stuff.
Yeah.
Little k's mean rates, big K's mean equilibrium.
Yeah, both the letters are like K is the most important.
Yeah, it's like K became the most important thing other than Q. But it has the K sound, so it must be a quantity or a composer. All right. So ultimately, the equilibrium constant for the association will be the product and the ligand together. Makes sense, right? That's the product. Things coming together over the individual concentrations of the protein and the ligand. Okay. And so that's also analogous to our Ka over our Kd. Setting it up. So far, so good. All right. So in practice, we can often determine the fraction that's being occupied. All right. Sometimes it's hard to measure exactly if all my protein is actually occupied or not, right? But I can figure out how much of it is under a given set of conditions that we've established. And so that's what theta is going to represent, is the fraction occupied. So if it's 50%, then that means half of it's occupied. If it's 75%, then 3/4 of it's occupied. That's what we're going to be looking at, right? Of the binding sites, and that's the key idea, because we're going to talk about what happens when you have more than one binding site today. All right, since Ka is equal to this, we've already established that, right? That would mean that if we substitute PL with K times L times P, so what is this right here? Just take this over here, right? We just end up taking what's in the denominator, moving it over. All right, if we take all that, then that's going to be equal to PL, all right? Then we can eliminate PL from the equation altogether, because we don't actually know what PL is, per se. And then we can get something that's a little more complicated looking, but what is it? It's the same thing for the most part. We'll just happen to take these and put them up there. So now that we have common factors in it, we can go through, do a few things by eliminating P, rearranging, gives us the result in terms of an equilibrium association constant. And this is what we see. Do you feel like life got better?
Yeah.
Still.
Yeah.
We're gonna go, all right, now we have a now we have a fraction in a fraction. That just doesn't look good. I agree, so let's go further. All right, so if we have this association, now we just look at it. Look at what it's in terms of. The ligand concentration, we know that, we put it in there, right?
Mm-hmm.
And then what do we have? Well, we have 1 over the equilibrium association. So if we can get that, then we'd be great. But guess what? By definition, the equilibrium dissociation constant is equal to Kd, the dissociation of equilibrium. So instead of having a fraction in there, we can take the Kd and shove it into that same spot. All right, so if we do that, we now come up with this. Can we figure out the percent occupied? We could do that.
Yeah.
We could figure out how much of our protein is actually bound with ligand. All right, there are different ways to do it. One way is you can have a radioactively labeled ligand, and you can find out how much of your protein, which if you measured your protein and put in there, you would know exactly how much protein was present with that ligand in it. And so you could do different concentrations and you can measure it. You can do it by some other techniques where you actually cause it to get stuck, to where you might do it by a gel shift assay, to where you actually have it, if it's big, if the ligand's big enough to make the gel run differently, the protein run differently. You can watch to see how it shifts its mass in a gel, potentially. There are other techniques, some are different other techniques you can use. So if we can just figure out what this is, and we know the ligand concentration, we can determine what our Kd is, our dissociation constant. All right, that's a big Kd, yes, the big one. All right, so now it depends on the free ligand concentration in Kd. That's what it basically, that's what we can worry, that's what we can imagine. So when we know all this, all right, so if you get the total concentration is approximately L equal to L, it means we're going to saturate the system, correct? And if that's the case, then we're close to 100%. All right, when you're at 1/2, let's say we're at half a fraction, what's going to happen? Well, we can rearrange it as such, and then what do we get? We get all of this. So what happens is, there turns out to be an inflection point in the behavior as to how the two things come together. And so if we go to half the maximum, in other words, this would be all the protein binding sites filled. This is at 1/2. We can come over here, drop down. Guess what that happens to be? It happens to be the Kd value. So interesting. This is a defining character of the association of the protein with its ligand. We can measure that, and then you can use this to actually say something about how effective does it bind. How much of it is dissociating at equilibrium, how much of it is dissociated compared to associated. All right, so that's what we see. So you can do this graphically, which is classical. You can go into the lab, like I was saying, you can measure a couple of the fractions, you can plot that, and then from there you can just basically drop down and determine what your Kd value is.
So just to clarify and get everything straight here, the idea is that any given ratio or any given theta, we can find a Kd value equal to that theta, that ratio of ligand we put in.
Close. So what I would say is, for any given ratio, we can plot that, and for a given protein, we could determine what the Kd value is.
Okay.
And then that becomes a characteristic of that protein ligand combination. So when you talk about a drug, for example, how much drug do you need?
It's going to be dependent on how fast it dissociates.
It is, and so what kind of a drug would you want if you're really out to make lots of money because you're an evil pharmaceutical executive?
It's the kind that people have to
You
take every
you want
day.
a low KD or a high KD?
Low.
High. High, so they buy more.
High, so they buy more? That's possible. Or low, so you can just sell them a very little with a lot of filler.
Very little.
Both are true.
Both are true. So now we can see that it's not good to be evil, because it's all about money. All right. All
Very
right.
nice.
So what it means, though, ultimately, is that we're looking at, you can actually characterize how it works. Now, if you really want a good effective drug, you want something that can outcompete the natural ligand in most cases. And so if you want it to outcompete the natural ligand, it needs to have a KD lower than the KD that naturally occurs. Because that's the dissociation constant. So what it's really saying is, we would have to put in less to get up to at least 50% or more occupied to block the binding of the natural ligand. That's what's called an antagonist, by the way. Okay, so if you're looking at an antagonist drug, that's what it would be. If you have an agonist drug, let's say you want to stimulate something, then the KD can still be lower, and then you can effectively stimulate that process by the agonist drug, the one that will actually affect the same result, but it's not the actual natural molecule. So that's what we're looking at. All right, so let's see, anything left to say there? What would you estimate the value of the KD to be?
1.
About 1. Looks like about 1. So, all right. So now let's really try this. X and Y. Which one is the more effective binding agent?
X.
More effective.
What's the evidence for that?
Lower
Should-
KD.
The lower the KD, right? The lower the KD. So we come here to 50%, drop down, and we find out that we're somewhere close to 2. Whereas with Y, we're clear out here at about 6. So you could say that Y binds 3 times less effectively. We were just to make a direct comparison between these two curves. All right. So those are the KDs. All right. So we have 2 and 6 apparently. All right. The lower the value, the higher the affinity. Don't forget this idea. When we come back and talk about enzyme kinetics next week, you'll see something fairly similar in the shape of the graph and everything, but it'll mean something slightly different, okay? Because you're actually dealing with chemical reaction being catalyzed rather than just a dissociation. All right. You often spend time on these problems in class?
No,
No, he just says to do that sometime?
pretty
Okay,
much.
do that sometime. If you have questions. I guess the office hours are typically in here at like on Tuesday, Thursday at 2,
Yes.
or whatever time we meet for class. Yeah, 2 o'clock. Is that true?
Yeah, I think he normally does it Tuesdays.
Just Tuesdays.
Oh, just Tuesdays?
Yeah.
Thursday's got another class or something, but.
Oh, yes, no, that's true, he does have another class on Thursdays. Okay, so at least tomorrow, I think I can do that because I think I have normal office hours during the time anyway. So I'll just redirect them to this location.
Okay.
So try this out, see if it makes sense. If it doesn't make sense, come back tomorrow. Double check my schedule. Yes, that should work. All right. All right, so there's some stuff. You can see the math. You can check it out. All right, so some examples of KDs if we're looking at things. So there's what is called avidin. It's in egg whites. It's actually it's turned into a biotech tool, by the way. All right, it binds biotin specifically. And not only does it bind it, but look at how well it binds it. What scale is that? It's like, okay, that's like, what? Billionth would be to the minus 9th. Trillionth to the minus 12th.
I don't know anything beyond femto.
Femto is it.
Yeah?
Yeah, well, it's not atto. Atto would be the next one, but. So this is on the femto scale. Now you might ask, why is avidin in egg whites binding biotin so effectively? And the answer is, it protects the egg from bacterial infection.
Wow.
It basically scrubs biotin out of the system. And you might say, what is biotin anyway, right? Anybody know? It's one of your B vitamins, vitamin B7. It's involved in a lot of carboxylation reactions, actually. So if you go take 42, highly endorse it, Anyway, that's the class you can actually take, and then you'll find out what biotin does. And so what's really fascinating is since it binds it so effectively, it works really well as an antimicrobial agent. But what's happened is people recognize this, so what can you do? You can stick biotin onto anything, and then you can easily have the avidin grab onto it, and anything attached to the biotin is now been selectively pulled from solution. So you might remember your affinity chromatography from chapter 3. You can have some kind of an approach. Often they'll either put avidin as a protein conjugated to the protein of interest, or you can put some kind of a linker with biotin on it, and you can selectively pull it out, or you can selectively identify it at very low concentrations. So it's very effective that way. All right, and so if we look at some other things, we have insulin. Insulin binds it like 1 times 10 to the minus 10. So you'll see there are a lot of things that happen over here in this 10 to the 9th range, 10 to the 8th range, somewhere in there. All right, that's where you tend to find a lot of your ligands. We'll come back and talk about enzymes and substrates, and you'll note that they are worse at binding. There's a good reason for that. That's what you'll start talking about next week, right? All right, so this is biotin, by the way, right here. Molecule itself. You see how it's bound in the avidin, the amino acids around it. All right, so let's go ahead and start talking about a case study for binding. We have hemoglobin. All right, we're going to be looking at oxygen and carbon dioxide as the two molecules that are involved. What can you say about carbon dioxide and oxygen as molecules? What was that, Haley?
They're small.
They're small. And what was the other one?
Non-polar.
The non-polar type things. So not very good to grab onto, are they? And yet it's essential that we have oxygen because...
It's how we live.
Can oxygen diffuse through your skin? Absolutely it can. It's non-polar. There's nothing blocking it, right?
You can't get it deep enough or in high enough concentrations.
There's the real problem. It does diffuse through your skin, but that's about all it goes through because then the cells consume it and it's gone. So getting it internal to your other organs would be the issue, correct?
Yeah.
Interesting enough, there's a book called Oxygen by Nick Lane. He talks about the fact that if the oxygen concentrations were higher ones, anybody ever see the dragonflies that are in fossil records, how big they are?
Yeah.
I mean, you're talking like dragonfly that's like huge. Insects don't have a circulatory system. Oxygen has to diffuse. So to have that large of an insect means that the oxygen concentrations must have been much higher for them to survive. So,
Thanks for open series.
interesting stuff that way. All right, let's take a
For all
look at
intents
this.
and purposes.
All right, so some red blood cell fact sheet, just see some things here. In other words, we've got a lot of red blood cells. That's really what you should take away, right? All right, white blood cells, not so many. Platelets, a lot of that though too, but mainly it's the red blood cells that are really circulating around in our system. All right, and it turns out that for each human red blood cell contains about 270 million of the hemoglobin molecules, each carrying 4 heme groups, or 4 binding sites for oxygen. So 4 times this times whatever that is up there, and that'll tell you how many oxygen molecules you could potentially be circulating.
Dang.
All right, hence it's a chemistry class. Probably better off to use something like moles. All right. All right, so these, they do, okay, so the red blood cells do develop in the marrow. You should know something about that. They circulate for approximately about 3 months, 4 months, somewhere in there. All right, and 1/4 of the cells are basically, essentially, in the human body are red blood cells. All right, so a few interesting things. Oh, this is important actually. So mammalian red blood cells are unique because they don't have a nucleus. It gets expelled. So these can't reproduce, they're just there they're one and done for the next 3 months. All right, so no DNA is present. They also do not have like cellular organelles such as the mitochondria either. So they survive off of glycolysis, if you want to put it in terms of that.
Oh, wow.
Right? Now, here's this is what we actually have for the binding site. We have what is called this porphyrin ring structure. You can see that there's some nitrogens around here. There's an iron ion that is bound in the middle. It's typically a +2 ion. All right, so this entire ring structure looks like a Pokemon character. All right, but nevertheless, it's not. All right, and what's going to happen is this is a planar structure, and we're going to have 2 areas where there can be additional binding or ligand connections, if you will. Technically speaking, when you're looking at something like this iron and all these lines coming off, this is where it gets really confusing. In the chemistry world, we'd say that anything binding to a metal ion is a ligand. It's spelled exactly
I know,
the
it's
same.
freaking dumb.
Well, be careful which one's dumb. Alright?
Metal
One
one's
claimed
cool.
it before the other. Alright, so. Alright,
It's
so
like we gotta, like-
ligands are these things that associate with an iron or some kind of a metal ion. Ligands are typically organic molecules that bind to a protein. So here we have a case of something called oxygen that's going to actually bind to the iron. So what is it? Is it a ligand or is it a ligand? Or do we really care? Alright? Just making sure you know that
Do I need to know
conflict
the
right there.
test?
That's
So you're gonna form like an iron-oxygen complex, is the idea? Okay.
You are, just like this. So if we have an edge-on view, so this is our porphyrin ring edge-on, so you're not seeing the structure of it. And just looking, oh, and there's a break here so we can see what's going on in the middle. All right, so we see that we have an iron, the iron ion's here. It's connected to a nitrogen that is part of what functional group here? What is that?
Is it a
Is it
imidazole?
imidazole?
It's a imidazole ring structure, so that would be our histidine, right? So our
Okay.
histidine from our polypeptide chain is going to link the porphyrin ring to the iron.
Wow.
Oh, okay.
Through the iron here, and then the oxygen is going to bind on the opposite side. Right? That's what's happening here. Now, what does it look like? It turns out that, so it's coordinated position with 93. So if we're looking at it, this happens to be myoglobin. What's myoglobin? It's in your muscles. What's its purpose?
Is that what contracts?
To make meat the color of meat. What is its purpose? It makes it reddish, that's true, but that's only because of the iron, but anyway.
To give
What
it
does it do in the
oxygen.
muscles?
Does it cause contractions?
It does not cause contractions, although it's necessary for contractions to happen. Because it's holding something vital for it.
The oxygen.
Is it just an oxygen carrier?
Yeah, it's a reservoir for oxygen. So not only do you circulate oxygen throughout your body, but we have the same consideration. If it might take too long for the oxygen to diffuse to the tissues, to deep in the tissues, therefore what's the solution? Put something there to store the oxygen so that you're basically just replenishing it at the surface and it works its way in, so it's not like the problems we've had over the past couple of years with Amazon. You order it and it says, okay, it'll deliver to you in like 3 months. Right? Supply chain issues, is how to solve them.
I have a question.
Oh, we've got two questions.
Just like myoglobin and red blood cells, they are different things.
Yes. So red blood cells have hemoglobin, myoglobin is going to be in the tissues. But this structure, this is the major point we make right here. This structure, notice this polypeptide chain and how it's folded. These tubes represent alpha helices. All right, and you can see that there are several of them. They've labeled them A through, what's the last one? F, G, and there's G that goes back there. H comes around here. That's it. Okay, so A through H. All right, so this is what's called the globin fold. So hemoglobin, myoglobin, they have the same structure. All right, and we're going to see a little bit about how this works. With myoglobin, it's just one subunit. It looks just like this. We have an iron here. We have two histidines around that porphyrin ring. This one's the one in close proximity, or the proximal histidine. This other one over here is distant or distal. Got those names so far?
Yeah.
These become important. These are actually something you want to pay attention to. You have your proximal and your distal histidines. Where do you think the oxygen is going to slide in?
Between the distal?
Between the distal and the iron itself. So this is the binding site for the oxygen right here. All right? That's where it's going to be. And that's true for myoglobin, and it's also true for hemoglobin.
So that's that porphyrin ring, right?
So this is the porphyrin ring, the red thing highlighted here. And it's associating with the protein just through non-covalent interaction. Right? It's just sandwiched in there.
Those names have to do with where they're located on, not which one's closer to carboxyl terminus and amino terminus.
Correct. It's which one's closer to the iron atom.
Okay.
To the iron right
What
here.
is that?
This one's
What's that right
proximal, close in proximity of, and the other one's distal, but still in line.
Perfect.
All right. So that's the structural motif for the globin fold. And so if we look at this, so here's an example of oxygen binding to myoglobin. So again, going back to our previous, we can measure our ligand, we have our KD, that kind of thing. So what happens is, as it binds, notice what kind of binding do we have? Would you say that's a low or a high KD?
Low.
Pretty low.
Pretty low
A
KD.
pretty low KD. So what
Pretty
this
high
means-
affinity then.
means a high affinity for oxygen. So when the muscles get oxygen delivered to them, the oxygen is going to go and bind to the myoglobin and be held there in place until what happens? Think about it. So the partial pressure of oxygen is our concentration of oxygen. And so as the concentration of oxygen drops, it really doesn't change anything for myoglobin until it gets pretty low. Then it releases that oxygen to the cells for use. In other words, to undergo, so the carbohydrates or other energy sources can be fully oxidized.
So this implies that you actually have a pretty low concentration of actual oxygen in your body.
No, it just implies that the myoglobin, well, in the gas form, yes.
Okay.
Because basically, if it's in the body, it's going to be bound up quite frequently, whether it's
Okay.
hemoglobin or myoglobin, yes.
Got it.
All right?
Okay.
Yeah, that's a good way to put it. All right. Why would it be important not to have oxygen wandering free?
Free radicals.
Super reactive.
Free radicals, exactly. Free radicals is the issue. As oxygen, it does need to diffuse through cells. It needs to get into the mitochondria eventually so that you can undergo the catabolism process for whatever kind of molecule you're breaking down. But in the end, that's also what generates damage to cells. All right. All right. So that's what we have going on. Interesting enough, how many of you have done this before? Stick your finger in a little clamp thing and it tells you what your oxygen levels are. Yeah. You thought about how it works?
Like,
Yeah, so basically it's a spectrophotometer. And what is it doing? Well, it's actually measuring at a couple wavelengths. You have 650, around 650, and the other one's over here about 925. All right? And what do you know about visible light? Visible light ends where? Oh, I want to find a class, I can tell. Visible spectrum goes from 400 to 700.
Okay.
Violet to red, right? So this is red light over here in the 650 range.
Okay.
All right, so if we're looking at this, the blue represents deoxy, all right, deoxyhemoglobin. The red represents oxyhemoglobin. Here's what's pretty fascinating about it. So your blood is red, but the absorbance of red light is really low when oxygen is bound.
That's because it's reflecting the red, right? Instead of absorbing the red.
It's not absorbing the red, is it?
Yeah.
So therefore, it must be absorbing what wavelengths? The other blue, green, that kind of light is what's being absorbed by your blood. All right, so the iron itself is absorbing the higher energy light, not in the low energy light. That's what you see coming back. Now, why do they measure out here? Because what you want is, because we don't see the oxyhemoglobin, but over here you can get an actual ratio that can be measured, showing you something out here in the infrared range. All right, so they use these two wavelengths to calculate the actual concentration of oxygen. So, little spectrophotometer that locks over your finger and shines light through it.
Cool.
And using slightly longer wavelengths also pass through much easier, by the way. All right. All right. Oh yes, carbon monoxide. That's a fun one. So what's up with carbon monoxide? Well, iron typically has the oxygen bound to it. Carbon monoxide is also a small molecule with only 2 atoms. The difference, though, is carbon monoxide is linear, and it's because of the hybridization, sp hybridization as opposed to sp2, right? And so what we see then is this one will actually bind in a direct fashion. That means it has better overlap of the electron density in this particular case, and it also has what's called pi back bonding, by the way. So you have pi bonds that are coming off to the sides, and those will actually be involved with some of the interaction with the d orbitals. So that's what makes it so strong to bind. Here's what's really interesting, though. If you want to make meat look better to the average person, look what you could do. Expose it to carbon dioxide. Is that good, though?
No.
No, that's another one of those.
As long
bad
as the
money things.
consumers don't know.
They don't know, it won't hurt them.
Wow.
Bad thought. All right.
Science.
No, that's called greed. All right. So now what's interesting about this is, notice this line right here. Do the steric effect of the distal histidine on E7. So remember there was that other histidine that's out here?
Oh, yeah.
That will actually increase, how do they put it in this sentence? The affinity is increased by 40 times compared to its absence, all right? So what happens is the distal histidine, we'll see a picture in a minute, you can see that it'll actually help to form an association, so it helps stabilize oxygen binding better. It also has a tendency to destabilize the carbon monoxide a little bit because it'll actually push it a little to the side so the overlap isn't quite as strong. It doesn't mean that carbon monoxide's not going to kill you. It's still 200 times more effective at binding than the oxygen is, but that's better than 20,000 times without it. Okay. That's out of the book, by the way. All right, so carbon monoxide binds well. All right, so we can see more about that if you want to look at the different things here. Yeah. So there's plenty there. Look at that if you want. All right, so here we go. So when oxygen binds to myoglobin, the iron changes position in the porphyrin plane. So this is what's really interesting about it. So notice the iron, when there's no oxygen down, it's actually not in the plane of the porphyrin ring. On the flip side, once oxygen does come and bind, it actually helps pull it in line. Now, what consequence does that have? Well, if we imagine this is connected to the protein, it'll actually pull this part of the protein in a little bit closer. By changing that position of the protein, we cause a slight shift within the structure of the protein. And so you might recall that sometimes you can have what we call a conformational change. This is how there's an induced conformational change due to the binding of oxygen. That's what happens with myoglobin. And we'll see the same thing happens with hemoglobin as well, but there will be a different consequence. All right, so the role of the distal histidine, it makes oxygen bind in a bent fashion. All right, but then the other thing that also happens is that we're going to have it pull out, so this is going to help with the ultimate end here. Oh yeah, this is an interesting fact here. So you have an iron +2, and that's good. The problem is if the oxygen picks up the extra electron, then it dissociates. You've now got oxidized iron, which does not bind oxygen well, and you now have superoxide iron as well.
Which would be bad.
That would be very bad.
Yeah.
All right, so oxymyoglobin, oxyhemoglobin. All right, there is evidence for this superoxide ion as well. You can actually, there are different techniques that can actually help to suggest this, different spectroscopy techniques specifically. If you're interested in that, they're really kind of fascinating.
So wait, why don't, are you saying that these superoxide ions do form in your body?
While it's bound, it's actually in that state, but if it dissociates and when it takes the electron with it is the problem.
Okay, wait. So it is in the superoxide ion state.
Well, it can be resonant between.
Okay.
Yes, it can be resonant. All right. Let's see. I don't know if there's any more I want to say about that. I don't want to get over here more. All right, so again, here's the structure we're looking at. So there's a hydrogen bond between the oxygen and the distal histidine. All right, so we see that going on. And the importance, of course, is this is how the information can be transmitted between binding of an oxygen and the rest of the protein. Why this becomes important? Well, in the case of myoglobin, it's not so important other than you have a loaded, unloaded state. But when we start looking at myoglobin and hemoglobin, this is where we start to see something different happening. Notice this is hemoglobin. It has 4 subunits that look just like the myoglobin subunit, but they're stacked together, so they actually have some kind of physical contact. And note what happened to the KD of oxygen. Yeah, the KD has shifted, right? Whereas before we saw something that was very low right here, we now see it somewhere clear out here at about 26 torr roughly is what they have the P50 at. So it's changed. So that means hemoglobin is worse at binding oxygen than myoglobin. Why would that even make sense?
So it can dump off oxygen.
So it can dump off oxygen. Its job is not to hold it, its job is to deliver it. Imagine for a moment, the Amazon truck comes right around your neighborhood, you're expecting that package. It has a high KD.
So just, they don't give it to you.
It's like the truck can either have if it's a low KD, it's going to keep it. You want something with a high KD, so they'll dump it, right?
Yeah.
Yes. Last thing you want is a delivery truck that doesn't deliver. It just fills up with all sorts of stuff. All right. And so it turns out then that it's the association between the subunits that's responsible for this increase in the KD, or in other words, the effectiveness of it being able to bind and release. All right? It can bind the oxygen, and typically where is it going to bind the oxygen? Let's think physiology here. Where does oxygen get bound?
In your lungs.
In the lungs. High concentration or low concentration of oxygen when you inhale?
High.
High. So it makes sense that it doesn't matter if it doesn't have a great KD, because the concentration is going to be much higher anyway. And that's okay. So you can still get it more or less saturated. All right. So on the intake, we can get it clear up over here. And then as it goes throughout the circulatory system, it comes near the tissues, and the concentration of oxygen starts to drop off, then it's going to shift back, losing the oxygen. And that oxygen can still go over and effectively bind to the myoglobin in the tissue. Right. So that's the basic plan that's happening.
I have a question.
Yes?
So does that mean though that it's more difficult for your body to effectively oxygenate tissues that are distant from your lungs, or does it matter?
That would be true. The evidence for that is limbs, when you're talking about cold. I mean, you might experience that, and the same is true if you get into high elevations. You might find it harder to feel your extremities, your fingers, toes, that kind of thing. So, yes, because and part of it is also because of the way the circulatory system is designed, that it goes from larger arteries down to smaller capillaries and such. So, all right, we'll talk more about that hopefully before too long. All right. Now, looking at this is really interesting. So the first column is myoglobin's amino acid sequence in each of these columns here. And then we have aligned with it what's called hemoglobin B, alpha, and beta. All right, so there are two different, so hemoglobin itself is a dimer of dimers. All right, so you have two alpha subunits and two beta subunits. And they'll associate, and you can see that, you can see the myoglobin with that versus the alpha versus the beta. These are alignments that are sequence alignments of the protein sequence itself. And if you look at it, what happens is we can note that we start out here, and then there's leucine that seems to be conserved, there's a valine that's conserved, tryptophan. The conserved ones are the ones that basically are boxed. All right, we can see this lysine, this histidine, and this histidine. There's our proximal histidine, there's our distal histidine.
Wow.
So they both have the same fold, but it doesn't mean they have the exact same amino acids all the way through. Yet, they're close enough, but there's still room for some variability, and it still works. It still folds in the same way. So it doesn't mean that you have to have the exact sequence to get the exact pattern. You just have to have similar sequence to get the same pattern. Okay.
For whenever they're listing, or whenever he lists the things that are on the exam, he says, so we need to know 3 histidines in hemoglobin.
3 histidines? Yes, we've talked about 2 important ones so far. There's
Okay,
one more
so there
coming.
is another one.
There's one more coming, yes. If we look on here, do you see any more histidines that seem to be important though? See any?
I mean, you have the proximal, the distal, and then there's one highlighted
Proximal, the distal, and then where's the other one?
on the end.
On the end over here. Where is it at? So we'll come to that here. Probably not today, but definitely we'll see. All right. So the sequence alignment tells us that we have something, and then the oxygen binding curve. So the way that this works is as follows. We can see very easily that the hemoglobin itself, if it didn't have this cooperativity, if you were to break the ability to have these subunits associating with each other, notice what happens really quickly. With no cooperativity, hypothetically, this is what it would look like. So what I mean by that is, when binding happens in one subunit, it communicates with the other subunits, facilitating that they can uptake oxygen more readily. So binding in 1 gets us a little further along. Binding in 2 gets us up to here. Binding in 3 gets us there, and binding in 4, that's where we are. All right? And so the fact that you see how it first starts out and it picks up, and then it goes across like this. So this pattern is what's indicative of cooperativity in binding for any protein ligand system. If there's cooperativity. So cooperativity almost always happens when there are multiple subunits that can bind the same ligand.
Is the no cooperativity line telling us about one subunit?
Basically,
like
this
each-
is how it worked as one single subunit.
Okay,
To
without
some
any
degree,
help.
that's the hypothetical. It turns out that if you actually get them separated away from each other, they actually almost behave like myoglobin.
Okay.
Which actually would make more sense. If they're completely dissociated, they would behave like myoglobin. So,
Oh, okay.
That's because
So, but anyway, so you can see there's a low affinity state, there's a high affinity state, and then hemoglobin actually just operates in between.
Cool.
So myoglobin, hemoglobin that doesn't have cooperativity, and then this is the reality of it. Now, what makes difference here? So if you're looking at it, this is the partial pressure in the tissues, and this is the partial pressure in the lungs. So basically in the lungs, hemoglobin is saturated. When it gets out to the tissues, you're operating more in this area about 60% saturation. So it's dumping off about 40% of what it's carrying
On the way.
on the way, which is good. Would you want it to dump everything when it got to the tissues? No, because you're one breath away from some real bad times. Wim Hof would not exist. You don't know Wim Hof is, you should look him up. It's actually very relevant to what we're talking about. So, anyway, not advocating what he does, I'm just saying it's really relevant. He talks about, how long can you hold your breath?
I've heard 2 minutes,
How long can you personally hold your breath?
A minute.
Maybe a minute?
maybe a minute.
You can do his silly exercise, and you can sit there for like a minute and a half, and you don't even feel the urge to breathe. It's really wild.
Wow.
I don't know, but it's a long time. And then he does all kinds of crazy stuff like, oh, this is actually useful. Yeah, we don't have much time left, so let's just go ahead and talk about this. So one thing that's really fascinating is the response to cold. What's your normal physiological response to cold? Shivering, but how do you breathe?
Pretty shallow.
It's pretty shallow. What's fascinating is this is counterintuitive because you're like, it's cold, I really don't want to breathe in cold air. But if you take deep breaths when you're like tried in a cold shower, just breathe deep a few times, just in a real cold shower. Within a couple breaths, you don't even really feel the cold now. And it makes some sense because what you're doing is you're still trying to force your blood flow to act normal, which distributes heat and oxygen, so you can continue metabolizing rather than going through the shivering process, which is a non-oxidative process to try to get heat generated. So, anyway. So ultimately, what does cooperativity look like? Well, we have two subunits here. When one binds, it's going to cause a conformational change that's going to basically make it so it's favorable for the other one to bind and become stable as well. So, and then the reverse would be true. As one loses the ligand, it would be easier for it to go reverse as well.
So are they trying to show us that it's like, it starts out as like half opened hands, when one closes, the other opens up more?
Well, some things where it causes the other one to favor binding something as well.
Okay.
That would be the idea. Right?
Cool.
All right, so with that, we will stop there because the next part is this weird model and stuff. We'll talk about the hill. We can start with the hill plot and then plow on through into the actual structural features. And then